Hello, my name is Iliana Baums. I'm a professor of biology at the Pennsylvania State University, and I have the privilege of sharing the CRC's genetics working group. Working group members are Andrew Baker, Sarah Davies, Andrea Bertoli, Carly Kinkle, Sheila Kitchen, Ilse Kuffner, Michelle Matz, Margaret Miller, John Parkinson, Carlos Prada, and Andy Shantz. The products of the working group very much represent a consensus of thought, only achieved after many hours of intense discussion. And I'm very grateful to all my colleagues for their contributions. I will first highlight an important issue that has recently come to my attention, the need for genetic management plans, and then present a brief update on our activities over the past few years. Our working group has been chiefly concerned with how to maximize adaptive potential of coral populations to a rapidly changing climate. As a group, we think it is critical that we maintain genetic diversity of coral species as the best bet for corals to adapt to a changing climate. Later today, Carly Kinkle will present our thoughts on how effective selective breeding might be to enhance thermal tolerance of corals and whether enhancing thermal tolerance alone is the appropriate goal when we're planning coral restoration and conservation efforts. As a result of our working group discussions on selective breeding in combination with the kickoff of Mission Iconic Reefs in Florida, I personally began to think very seriously about the need for genetic management plans. This is because just 5 years ago, it seemed unlikely that whatever we do would be enough to change the real frequencies of coral populations, say across the Florida reef tract. And yet I believe we're now at that juncture. Why do I bring up changing allele frequency? Well, that is because we define adaptations of change in allele frequencies caused by selection. Because we used to have so many millions of coral colonies, it seemed impossible that we'd be able to change allele frequencies in the wild. However, in Florida, we're now talking about a habitat with very few corals and a project that will add tens of thousands of colonies to this habitat and hopefully be successful in doing so. So changing allele frequency is now on the table, and I began thinking about ways that we can prevent unintended erosion of genetic diversity, especially in coral breeding. Restoration projects are increasingly relying on captive coral breeding to generate new colonies made possible by incredible advances in coral reproductive biology. One of these innovators is Val Chamberlain, who created this figure and is a co-author on a manuscript we recently wrote on the topic. Wild and lab-bred coral larvae both experience very high levels of mortality during development, and we now have data on how that reduces genetic diversity. What we have found is that captively reared coral larvae experience severe genetic bottlenecks from the time eggs are fertilized to the time young corals are outplanted to the reef. However, here's an example of the cumulative effect of these bottlenecks on restored populations of Acropora palmatum. Consider the first row of this figure produced by Nick Locatelli. What is shown is the pairwise kinship between 11 coral colonies that were reared in captivity in 2011 and are now grown on a reef in Curacao. The lab-bred cohort is sexually mature and spawns every year, which is a great success for coral restoration. As you can see from this genome-wide data, 11 coral colonies are closely related. They're siblings, half-siblings, and cousins. This is even though they were created from a batch of 7 unrelated parents. Now, if you compare this to wild coral colonies from natural reefs, you can see that this is a species that usually disperses far away from its natal reef, so we never find siblings or half-siblings or even cousins in the same reef. Thus, outplanting a highly related cohort of lab recruits on the same reef led to an unintended erosion of genetic diversity unbeknown to the practitioners. What we need are genetic management plans that let us manage the genetic bottlenecks during captive rearing and use these valuable coral recruits in the best way possible. For example, these plans might lay out guidance on how do we outplant these recruits across reefs to avoid future inbreeding between the F1 generation and wild colonies, as well as their F2 offspring. Our working group has produced 4 papers since we got together to provide hands-on advice for coral restoration and conservation already. We held a meeting in 2020 to identify our future priorities as a working group and decided to work on 2 manuscripts: how to manage expectations for selective breeding of supercorals and how to facilitate high-throughput phenotyping of coral colonies. We further collaborated with other CRC working groups and outside organizations and conducted various outreach activities. I'm grateful for the efforts of my colleagues in generating these ideas and it has been a privilege to share some of these thoughts with you today. I thank you for your time. If you are interested in any of these topics, feel free to contact any one of the members of the working groups.